From Microbe TV, this is TWiP, This Week in Parasitism, episode 144, recorded on December 11th, 2017. Yeah, you're right, it's gross. Hey everybody, I'm Vincent Racaniello, and you're listening to the podcast all about parasites. I don't say that at the beginning. You threw me off my game. Joining me here today in New York City, Dickson Despommier.
Hey there, Vincent.
What's it like out?
You know what? It's actually kind of nice.
Good.
There's not a lot of wind. There's
We had snow.
Fluffy little clouds out there.
We had snow this weekend.
We did. Yeah,
Also
we did.
joining us from remote location, Daniel Griffin.
Hello, everybody.
Did you have snow out there in the island?
Oh, we had about 5 inches on Saturday.
Ooh,
it was
whoa.
great,
Wow.
except I forgot to cover my boat, so it filled up with snow. And then Sunday morning, I was digging it out.
Oh, you mean it's still in the water, or is it sitting on land?
So it's sitting on land, and we tried to sail on Sunday. And we got to the edge of the dock, and we're about to put the boats in, and some people came over. The dock master, he's like, I don't know. It's gusting up to 30. I'm not sure. I'll
Right.
come swam with you.
That's true. That's true.
I'm
That's
like,
true.
It's all good. It's going to be great. So we didn't quite get in before they put an end to our Sunday sailing.
Ah, so you would have gone in at 30, huh?
Sure.
Yeah, you and I might be doing this together, Dickson.
Speaking of remote locations,
Yeah,
that's
really.
right. You would have been somewhere in the Azores.
Well, so what I think actually got them was I was going to be sailing with my cousin, and in one of the other boats was going to be my 12-year-old son Barnaby with his buddy Luke, who's also 12. And I think that was what they said. I don't know about putting 12-year-olds. We're like, we're fine. We'll keep an eye on them.
The British did during the
Sure, yeah.
Revolutionary War, cabin boys.
Oh, these guys would have been fine. They're excellent sailors.
sure.
And we have crash boats there, and they're dressed appropriately, and seconds, and they're back out of the water. But no, so we didn't sail, but beautiful. I mean, snow everywhere. It just it feels like Christmas, right?
Indeed.
Well, it
Got
is
that.
December.
Yeah.
But bum.
Bum, bum. All right. Speaking of Christmas, let's get back to our TWiP case from 143, Daniel.
Yes. Let me remind everyone, or for those tuning in for the first time, acquaint you with the case. This is a case from the 1990s. It was seen by a colleague. This was a boy in his late teens at the time who initially presented to an emergency room in the United States with a chief complaint of visual disturbances and itching so severe that it was preventing him from sleeping. He had immigrated from Oaxaca, Mexico, and we talked a little bit about where that is. Mexico goes down, kind of narrows, and then bends up, and so it's right in that area after it bends up. So I guess Puerto Escondido is a little surfing town right there at the tip of the bend.
Are you talking about the Yucatan by any chance?
So you're almost to the Yucatan. So you're between the bend and you're not quite to the Yucatan, and you're in I think it's Montalban is one of the ruin sites that's right there in Oaxaca. Anyone who's never been to Oaxaca, it is beautiful. But this young man was living in a less than affluent part of town. He was living in a modest dwelling with dirt floors. He had no running water, so he would go to the local river to get the water for the family. That was his chore. He reports that there were dogs, farm animals, many insects. We jumped to exam, where it was noted that he had tender nodules on his head. His skin was irritated from scratching. They were concerned about the vision issues, so an ophthalmologist was called in and reported that there were small punctate lesions on the right cornea. And then he gets referred to further specialists for diagnostics.
All right. We got lots of guesses because we're giving away another book.
Exactly.
The first one is from Wink. Dear TWiP professors, if you search the text in capital letters for cornea, you find Onchocerca, Trichinella, and Thalassia. However, my guess will be different, and I'll probably be wrong, but I've gotten used to that. This case was from a while ago, and long ago I participated in some field clinics in Panama where the Gorgas course was given there. We often saw scabies, and in the San Blas Islands, the mite burden was particularly high. I wonder if scabies can sometimes affect the cornea. or if persons with heavy burdens of scabies will scratch their own cornea while relieving an itch near the eye. Love your whole podcast, every issue. Wink from Atlanta.
Cool.
Dickson.
Daniel writes, and that's not our Daniel, that's someone else's Daniel. Dear TWiPtomaniacs. Now, that's the first time I've ever heard that one. Fresh from my randomly generated victory 2 episodes ago, I'll take another guess. It is in the unlikely event that I win another book, I'd like to donate a Spanish edition to the Hospitalito Italian.
No, Atitlan.
Atitlan, I'm sorry, Atitlan. Lo siento mucho. In Guatemala, if they want or need it. Otherwise, please send it to the good cause of your choice. I had a great time volunteering there many years ago. The fact that we have to go way back to the 90s makes me think we are dealing with a disease of historical interest in that part of Mexico. The combination of intense itch, subcutaneous nodules, and punctate keratitis in someone living close by a river suggests the patient has onchocerciasis or river blindness. This neglected tropical disease is more commonly found in West and Central Africa, but there are foci in South and Central America. The causative organism, Onchocerca volvulus, is a filarial nematode transmitted by black flies, Simulium species, which breed in fast-flowing rivers and streams. In fact, I just have to add a little thing here. They are the best filtering organism that lives in fresh water. They filter like crazy, and they have a wonderful system for doing so. So they're really, really, really good at taking advantage of small particulate organic matter, which is the ecological equivalent of their food. The symptoms, now back to our story. The symptoms are caused by an inflammatory response to larvae circulating in the subcutaneous tissue and in the eyes. Treatment is with ivermectin every 6 to 12 months. Ivermectin kills the larvae, but not the adults, so needs to be taken for the lifespan of the adult worm, 10 to 15 years. It is still a common cause of blindness. After some Googling, I found that a focus of onchocerciasis transmission did exist in Oaxaca. It looks like it was eliminated by 2009 as part of an ongoing regional program of ivermectin mass treatment. Best regards, Dan.
Dickson, when you read, your voice goes down, but when you converse, it's excited as usual. Is that because you're concentrating?
Perhaps. I can't multitask.
Oh. All right, Daniel.
So I was just going to say to the other Dan, so it's odd to someone else has your same name. As far as Hospitalito Atitlan in Guatemala, if you send us an address and a contact there, we'll work with trying to get them Spanish editions.
Indeed.
So just let us know. We'll still send you yours should you win. But yeah, the more places that need our books that we can somehow get them to those folks, then let us know, and we're going to keep doing that.
But of course, they can already download a free PDF Spanish version.
Version.
I meant version. Did I say version?
You said something else. But okay. Hello, TWiP team.
I've never been read before.
I've been listening for at least a year, but never worked up the courage to submit a case study response. Here goes my attempt. The visual disturbances described in case study number 143 initially brought a few possible parasites to mind. Ophthalmic cysticercosis from T. solium, which could cause visual disturbances, but does not explain the itching or the described tender nodule. Acanthamoeba keratitis should also be responsible for deteriorating vision, but again, does not explain the itching or tender nodule. Additionally, the rare cases of this disease are generally associated with contact lens use, which was not mentioned in this case. Ocular larva migrans could explain the vision loss, but again, does not explain the other symptoms and is more common in younger patients. Onchocerciasis caused by the filarial nematode Onchocerca volvulus fits all the described signs and symptoms. Although now considered eliminated in Mexico, thanks to treatment of affected communities with ivermectin, it was known to exist in Oaxaca, the patient's hometown, in the 1990s, and can persist for years in the host, meaning it could have been acquired years prior to the onset of symptoms. The Simulium species black fly is the intermediate host of this parasite. L3 larvae are spread from the black fly to the human definitive host while the fly feeds. Inside the human host, the L3 develop to adults and form subcutaneous nodules from which they release microfilaria for an extended period of time, years. The microfilaria hang out in skin, lymphatics, etc., until they're ingested by a black fly. They penetrate the midgut, where they develop into L1 and through to L3 larvae, before migrating to the mouth parts, ready to infect another definitive host. It sounds as though the disease is in its early stages, but there's only one Onchocercomata that we know of, and no other symptoms of microfilarial migration have occurred. Diagnosis can be made by observation of microfilaria from a skin snip, treatment with a single dose of ivermectin to control migrating microfilaria. Because this doesn't kill adult worms, surgical removal of the Onchocercomata may be beneficial. If surgical removal is not performed, a repeat dose every 6 months for as long as the adult worms are producing microfilaria, which could be years, would be required. Antibiotic treatment with doxycycline may also be helpful, as Wolbachia are a known endosymbiont of O. volvulus and seem to be required for a number of necessary parasite biological functions. Thank you for all the work you do. I've learned a lot from you. Perrin, and he's from Guelph, Ontario, Canada.
So where can you find the Onchocercomata?
Onchocercomata. Yep. Sure, you can just palpate and find them. They're actually bumps.
Where would it be?
It depends on where the insect actually built.
So it could be anywhere on your skin, basically.
Yeah, but in Central and South America, because they usually wear clothes and their arms and necks and heads are exposed,
I see.
You'd find them mostly around the head. And therefore, the blindness in those countries are higher than they were in Africa because of the less clothing worn by people in Africa. The flies could bite you anywhere.
Anywhere, I see.
And then the microfilaria doesn't have bigger.
So if you take out the
the nodule.
Hey.
Onchocerca nodule.
Onchocercoma.
Onchocercoma. Then you don't have to treat for a long time with ivermectin, right?
No, ivermectin kills the larvae, remember. So yes, they can still stay alive for 6 months after you take out the adults. So you should still treat.
Yeah, but that is an interesting thing that Dickson, Vincent, you're bringing up. So we do now know that removal of the Onchocerca nodules is beneficial.
Mm-hmm.
Yes.
And so you want to do that because what are these? It's sort of a nest where you've got the adults and that's where the microfilaria are being produced.
Right.
The ivermectin won't kill them, so surgery can. Maybe the doxycycline can. So that changes things a little. But it is an interesting question. If you remove the Onchocerca nodules, are you all done? So, you know, as Dickson brought up, you get rid of the Onchocerca, you give them their ivermectin. Are you done? Or do you need to keep treating them every 6 months? You're not sure.
If they move away from the endemic center, then yes, perhaps. But if they stay there, then they could be reinfected.
And actually, no. That is the critical. You know, I guess we're talking about Onchocerca, so we'll talk a little more. That's the critical distinction in the treatment: is are you still in the endemic area? If you're in the endemic area, these people just keep getting treated every 6 months for years.
Right.
But if you're outside the Onchocerca area, most of us, I would say, wouldn't just treat them once and call it a day, but we may not treat for that much longer.
Right. We'll come back to this because there's
we will.
Stuff to talk about.
The microfilaria never become adults in the human.
No, they do not.
They have to go through the fly.
That's right. It's a necessary developmental stage.
Yeah, it's good then for treatment.
Absolutely.
All right. The next is from Dave the Sheep Shearer. Good doctors. It's been a while since I submitted a guess, but here goes. The young man from Mexico with eye and face problems. When I Googled parasites, eyes, Mexico, I got 3 possible choices. Toxocara, however, the worm heads for the back of the eye, and the motile worm under the skin is a longer single worm. The timing of this is important. The 6th edition says that Onchocerca volvulus was recently wiped out in South Central America. However, in the 1990s, there may still have been cases in southern Mexico. It also says that in Africa, the cutaneous form of the disease is usually on the lower parts of the body. However, in the Americas, it's on the upper parts. The description from the 6th edition looks very much like what was described. Now, here's a novel approach. I would ask the patient if he had seen this before or knew what it was. If it's river blindness, it's likely that he has seen it before and knows or is familiar with it. I feel that asking a patient who has recently immigrated if they have seen this before can at the very least tell the doctor how to approach the patient. In your case of the man from Africa with the ulcer on his ankle, asking him if he has seen this before or is familiar with it could tell the doctor how to proceed. If it's a lot different breaking it to a born and bred New Yorker that they have a meter-long worm in their leg that has to be slowly pulled out. However, the man from Africa could be familiar with the procedure. It's great. So here's my guess. Onchocerca volvulus. The treatment mentions the use of ivermectin in a single oral dose every 6 months for the lifespan for the adult worms, which could be up to 17 years. It also mentions surgically removing the worms. I wait with bated breath for your talk on Lyme. My symptoms go back 3 years, and I was likely reinfected every spring as I worked my way through the Okanagan area of BC shearing alpacas. I've been unable to work since April and have been through the roller coaster that is Western medicine and Lyme. Dave, the sheep shearer from southern Alberta, where it is -4°C and calm. All right. Sorry about the Lyme. We'll do a Lyme episode soon, and we'll do one, and Daniel will have a Lyme case to kind of kick it off, right?
It should be on TWiP, however, don't you think? Yeah. Should do that one on TWiP.
Yeah, I guess that would.
You think that's a TWiP?
I think it's a TWiP.
For sure.
Yep.
Yeah, but you're not on TWiP.
No.
We could do a TWiP by ourselves without the other hosts.
We could do that. It wouldn't be wrong.
I can do it.
No, I would like to participate in that. That would be great.
All right. Dickson, can you take the next?
Alicia writes, Hey, TWiPers. This week's case description initially really threw me off, but I think I have a good guess after looking into it a bit further. So the symptoms of this patient immediately made me think of onchocerciasis, aka river blindness. Then I thought back to the location description, though. Oaxaca? Not Africa? After pulling up an epidemiological chart of the infection, I was met with more befuddling info. The infection seemed to be indeed centralized in sub-Saharan Africa. It was only when I read the history of onchocerciasis that I found that Oaxaca and other southern Mexican regions were once active foci for onchocerca volvulus infection. Starting in 1992, however, the Onchocerciasis Elimination Program of the Americas, OEPA, sought to eliminate transmission in these areas. Moreover, according to the World Health Organization, as of 2016, this program has succeeded in eliminating transmission in 11 out of the 13 original foci. The two remaining regions lie within the southern Venezuela and northern Brazil, respectively. Now, with this knowledge in mind, since this case comes from the 1990s, onchocerciasis becomes a legitimate diagnosis. When an individual is infected with onchocerca volvulus, the larvae begin migrating through the skin, eyes, and other organs. As they die, they elicit strong immune responses that could explain the itching of the skin and the keratitis in this patient. Additionally, if the worms reach adulthood, nodules can form around them, another symptom of this individual. In order to get a more confident diagnosis, skin biopsies should be taken and examined under a microscope. I believe PCR and immunofluorescence techniques are also available. The treatment approach for onchocerciasis is a once or twice yearly dose of ivermectin for 10 to 15 years. Why so long? I asked myself after reading this. Well, it turns out that ivermectin is able to robustly kill and prevent the release of the larval worms in these individuals, but the adult worms are simply paralyzed temporarily while existing in the nodules. These adult worms then continue to live on and produce progeny despite the recurrent ivermectin doses. Therefore, the lengthy treatment duration is warranted. Regards, Al. I must interject a note here. If you call the skin snip a biopsy, they will send it to the pathology lab. They will preserve it in formalin, and then they will section it, and they will send it back to you. Whereas if you call it a skin snip and then make an impression smear on a slide, the larvae that are still alive will actually come out and be stainable with Giemsa, which you can then do an examination under the microscope and spot them that way.
Neat.
So I would suggest calling it a snip rather than a biopsy in this case.
Dixon, while you're saying that, you want to talk about the blood-free aspect of doing these snips?
Oh, yeah, because if you dig a little deeper, then you're going to confound that. When you make that smear, you're going to have lots of confusing artifacts to have to put up with. So you have to lift it up with a needle, so you make a little tent of the skin, and then just underneath it with a razor blade, slice it just under the point of the needle. And that will give you a blood-free snip, as they would call it.
A blood-free snip.
There you go.
That is a great title for
I'll
this
have
episode.
I'll have a blood-free snip, thanks.
What would you like
that
with that
with or
snip?
without ice?
Right.
That's right.
Can you read the PS? Because I can answer.
Oh, yes,
the
P.S. Maybe I missed this announcement in an earlier episode, but was there mention of creating a This Week in Immunology podcast? If so, neat. Are there any updates on this progress? If not, darn. Thanks anyway, guys. Update.
Yeah, sure. We made one. We're on episode 2. Tomorrow we're recording episode 3.
Cool.
It's at microbe.tv/immune. So we decided to call it Immune instead of This Week. I want to get away from the This Week.
You should have called episode 1, episode TH1, and
Episode TH2. Right. Yeah, but we couldn't go any further.
No,
than
this is all true.
Good.
It's just 2 episodes. That's it.
I should actually announce podcasts on other podcasts to cross-promote.
Absolutely.
Microbe.tv has all of them.
You know, if you put a schedule up and then just embedded it into the TWiP family or Twix family, that would be a great idea, actually.
Yeah, but sometimes we're not.
Be a great idea.
We don't have a strict schedule, so to speak.
Not a strict schedule, but
All right.
least
Daniel,
the
you're next.
Okay. Melissa writes, hello, Twipsters. Woohoo. Thank you so much. I actually don't have a copy of Parasitic Diseases. I've been using the online PDF, which makes for searching for keywords easy. I'm glad that EH&S doesn't scare everyone. I never dreaded when EH&S stopped by Dr. Pride's lab. Actually, sometimes our lab auditor would stop by the lab to hang out, chat, and kill some time before his auditor later in the same building. That's how we got the inside scoop of what was going to be rolling out soon, so we could start taking inventory or get things queued up to get updated so we were in compliance. My guess for TWiP 143 is Onchocerca volvulus. I actually skimmed through the Parasitic Diseases book and looked at the photos to see if there were any with an eye drawn or photographed. According to Parasitic Diseases, to diagnose this, you need to get a skin snip from the area that is thought to have the highest concentration of microfilaria and put it in saline. Within 10 minutes, it should be looked at under the microscope to see if there are any microfilaria. The book also says that in Central America, the skin snip should be taken from the upper part of the body, which is consistent with where the patient is itchy. According to WHO, Onchocerca volvulus is transmitted by black flies, and black flies breed in fast-flowing rivers and streams. The patient could have been infected when he was getting water for the family. Parasitic Diseases says the treatment for this patient is ivermectin, and with a single dose every 6 months, it can slow the progression of the ocular and cutaneous disease. Thanks again. Melissa Lai, Associate Biosafety Officer at the University of California, San Diego.
How could we possibly disagree with Melissa? She quotes us profusely.
Yeah, that's right. Everyone does. Melissa,
no, I thought those were some nice comments that she made. I'll say that. And Dickson, you brought this up too. It's interesting, right? You know, in all the photos you see, I'd say all the photos. In a lot of the photos you see the nodules on the head, right? I think that's sort of a classic photo that's probably your fault, right? It's probably circulating parasitic diseases. But the location of these nodules is different in different parts of the world.
It's true.
And I like your explanation because, you know, how did Onchocerca volvulus get to the New World? Is it a different Onchocerca? Did it evolve here? No. It was brought. There was a period of time when, a lot of people from Africa would get free trips to the United States.
Free trips.
One-way
trips.
Trips against their will.
And much against their will.
Yeah, and that was, and this was one of, we'll say, sort of the consequences of that.
That is
so it's the same organism. So there's, cultural, genetic, there's always various things that affect why we see it one place versus another, but it's not that the parasite's different.
Right. This in Central and South America, it's usually associated with coffee growing, by the way, because coffee growing takes place on the hills where these streams are actually coming down, and it's a disease of coffee growers. So Juan Valdez, if you recall.
Do they have a name like Chicleros?
Yeah, no, that's a different one, but it's close. You're in the right ballpark at least. So it skipped from the African slaves over to the black flies, which were different species in the New World than they were in the Old World.
Yeah.
But they were perfectly capable of transmitting this infection, which is unfortunate because in most cases with these diseases that are vector-borne, the vectors are rather specific for a geographic region, like African trypanosomiasis versus American trypanosomiasis. But in this case, there was a biological cognate between the genus Simulium, which allowed for the wider spread version of it, unfortunately.
So now we have two people who have won books previously.
That's a good sign.
Again.
That's a good sign.
So what happens if they win again?
Well, lucky.
Yeah.
Hey, that's the luck of the draw.
All right, the next one's from Alan. Estimados profesores TWiP. That's nice. Your case study of a patient in Mexico in the 90s with tender nodules on the head, skin irritated from scratching, and small punctate lesions on the cornea sounds almost unquestionably like onchocerciasis or river blindness caused by the parasitic worm Onchocerca volvulus. The scratching would be typical of the severe urticaria caused by microfilaria passing through the skin, and the tender nodules would be typical of those caused by adult worms. And, of course, this disease is still the second greatest cause of blindness worldwide. Confirmation could be made from finding microfilaria in skin snip biopsy or seeing them on a corneal examination or excising a nodule finding adult worms. Treatment today, ivermectin periodically until the adult worms die. Some would recommend doxycycline to weaken the worms by killing their Wolbachia. We frequently would see onchocerciasis in mountains of Guatemala and southern Mexico in the 80s, but as of 2015, WHO had declared the region free of the disease. Thank you for another great podcast. Should I win the book, it will be a shared resource in the infectious diseases section of our primary health care courses.
Oh, great. Great.
What is the primary cause of blindness in the world? He said, onchocerciasis was the second. And Daniel, what would be the primary?
I believe it's vitamin A deficiency.
Yes, that is.
I like when I get things right, and it congratulates me. No, it's interesting. Here in Port Washington, we're the site of the Helen Keller Institute, which is really a tremendous organization that is not just local, but it's a worldwide organization focused on many causes of blindness. And for a very long time, chlamydia, trichomonas, eye infection was one of the top three. We're doing better with that. River blindness was one of the top three. We're doing better with that. Vitamin A deficiency continues to be a major problem.
Yeah.
So one of the issues is that people will use a grain like particularly rice as their main nutrition source, and it does not have vitamin A in it. And so they don't have access to, we'll say, vegetables, different things that have vitamin A. And as a result of the deficiency, there are hundreds of millions of people blind from vitamin A deficiency.
Wow.
So tremendous issue.
What's a common source of vitamin A that we would recognize in a vegetable that you could?
Carrots. Right?
Carrots. Okay, carrots.
Yeah.
Okay, beta carotene, of course.
There you go.
There you go. Okay, Todd writes,
Dickson.
The unsolved case study for TWiP 143 is an aberrant nematode infection of Toxocara canis. T. canis adults live in the small intestine of dogs and cats. Eggs from the feces embryonate after 10 to 20 days in the soil. If the embryonated egg is ingested by a suitable host, the larvae penetrate the intestinal tract and travel through the liver, bloodstream, and lungs and are swallowed to mature into adult worms in the gut. When the eggs are ingested by man, the larvae hatch in the small intestine and penetrate the intestinal walls and wander throughout the body without completing their life cycle. The patient's itching is most likely caused by a visceral larval migraines infection as the third stage larvae wander through the body and invade the tissues. The most severe forms of infection involve the eyes and other organs such as the heart and brain. Infection is most popular in children and adolescents under the age of 20.
I don't know if most popular is
well,
that,
yeah,
right?
most.
Let's try prevalence.
I would substitute prevalence for popular. That's true. Since T. canis eggs are not produced in humans, a definitive diagnosis can be done by histopath evaluation of infected excised tissue. There is a less invasive ELISA using serum that detects antigens that are secreted by the second stage larvae. Visceral T. canis infections can be treated with albendazole or mebendazole. Keep the interesting paper discussions and challenge cases studies coming. Todd. Yeah.
All right, we have a little divergence from the
We do.
mean here, don't we?
We actually do.
Okay, Daniel.
Peter writes, Dear TWiP Triptych, I am a postdoctoral fellow being mentored by Professor Celia Holland in the zoology department at Trinity College Dublin, Ireland. When first hearing last week's case study, I first thought of ocular toxocariasis due to visual disturbances, itching, and the presence of dogs. However, a quick consultation revealed nodules on head and punctate lesions did not sound right for toxocariasis. Then the symptoms of itching so bad that he could not sleep brought a talk to mind that I was lucky enough to hear by Nobel laureate Professor William C. Campbell just the previous week. Professor Campbell spoke of itching so bad that this symptom alone would warrant the need for treatment. The itching was caused by the disease onchocerciasis, also called river blindness, which would explain damage to the eye in the case study. I consulted parasitic diseases to confirm it, and indeed, nodules on the head were a symptom more associated with Central America than Africa, where the nodules are more often found on the lower parts of the body due to differences in the vector behavior and styles of clothing in the different areas. Punctate lesions were also more in keeping with onchocerciasis than my earlier diagnosis of toxocariasis. Finally, as Professor Campbell also showed a slide showing where onchocerciasis had been eradicated, I checked the information on Mexico. A WHO report dated October 14, 2015, verified Mexico free of onchocerciasis. So, the earlier date of the case being in the 1990s confirms travel history in an endemic area. So my diagnosis is onchocerciasis as a result of infection with Onchocerca volvulus, Lukart, 1893, via the black fly vector Simulium species. Definitive diagnosis by examining a skin snip and long-term treatment with ivermectin at least every 6 months. I hope I am correct, but after hearing Dickson saying not to remove his error from the last episode, I am not so fearful to put my attempt forward. Really enjoying hearing Dickson say not to edit it out. It takes someone very secure in themselves to allow it to be shown that infallibility is not a requisite to being a successful scientist. Very comforting for an early stage scientist like myself to hear. Professor Campbell took part in many functions while in Ireland, but one was an informal talk with TCD zoology undergraduate and PhD students and postdocs in his life in science and art that left us all inspired and driven to bravely pursue our curiosity without fear and to persevere. He also gave an equally inspiring discourse lecture at the Royal Irish Academy, where among many things he championed random empirical screening and hopefully helped me solve this week's TWiP case study. Professor Celia Holland gave a compelling response to Professor Campbell's discourse using Professor Campbell's journey in science to highlight the importance of mentorship for young scientists, the challenging of tackling neglected tropical diseases, the need for funding not only at an international but also at a national level to tackle NTDS, and finally, what an amazing world parasitology is to introduce young people into. On that note, I would like to thank you all for your work on the podcast. My bachelor degree in applied ecology, but my postgraduate and postdoc work have been on parasitology in wild mammals. And I find the podcast excellent for exposing me to new aspects of parasitology, particularly clinical parasitology in humans. I also have recently moved into the field of eco-immunology, so benefit from TWiM, TWiV, and Immune podcasts now also. I am lucky enough to be able to drive Professor Campbell around, and he was kind enough to sign my copy of Trichinella and Trichinosis. Who doesn't love a book with the line, Herbst was in the habit of dissecting various animals and feeding the remains to his pet badger. Through the book, Dickson came up, and when Professor Campbell asked, did I know him? I had to confess only in the way you sometimes feel you know somebody who has never met you when you have listened to them talking a lot on the radio through the podcasts.
Oh, that's very nice.
Apologies, the email is so long from Dublin, Ireland, where in keeping with the season, the wind goes right through you. Temperatures 3 to 4 degrees, gusty northwest winds. All the best, Peter Stewart, Trinity College, Dublin, University of Dublin.
May it forever be at your back.
So we in a small world, interesting enough, a lot of the coaches that come over here to teach sailing in the summer come from Trinity College, Dublin.
How about that?
So if he asks around, they probably know me in a different context.
I'll be darned. Did James Joyce reside there when he was writing?
Oh, does anyone know this? No. I know when we went to visit, we went to that where the book opens up, where
Bloom.
Yes.
stately
and
plump. Remember
and
that?
we swam in, what was it, the Snot Green Sea?
Yeah.
Yep.
He went to University College Dublin.
Right. But where did he? Then he went abroad. I presume he was an expat for a while, right?
I don't know. I'm not
Okay.
a
Joycean.
Joycean.
All right.
I can't read his books.
Well, they're pretty unreadable.
We have one more from Chris. Dear Twipsters, it's wet and just above freezing in Athens, Georgia. But amazingly, there's also a chance of snow. We may yet get a dusting before the year is out. This case of the boy suffering itching and visual disturbances brings to mind a few different parasites. Zoonotic infection can sometimes produce these symptoms. Toxocara larvae can migrate to the eye, although typically not the cornea. Cysticercosis can produce sensitive subcutaneous nodules and also affect the eye, though the presentation is unlikely to be the punctate lesions described. The most likely cause of these symptoms, however, is Onchocerca volvulus, which is endemic in Oaxaca. One report puts 500,000 people at risk of infection in the Americas. The patient's need to collect river water exposes him to the habitat of the black fly vector, and the ocular involvement is characteristic of river blindness. Mass drug administration has been conducted in Mexico since the 90s, so hopefully the risk is substantially less than when this patient was seen. The ideal means of diagnosis is observation of microfilaria in a skin snip. Adult worms can potentially be obtained from biopsy of the skin nodules. A sensitive but nonspecific filarial antigen test can also be performed. The current recommended treatment is ivermectin and doxycycline. Ivermectin should have been available by the time this case was handled and is much preferable to DEC, which can cause serious side effects. As always, thanks for the very interesting case and the reminder that these parasites still exist close to home.
Indeed.
Well, that was lovely, everyone.
It was. Amazing who comes out when there's a free book at stake. But that's good. That's just fantastic because we love giving them away.
We had 10 guesses.
That's amazing.
All right.
And only one that didn't actually pick Onchocerca as the definitive
That's right.
diagnosis.
So, Daniel, you feel good about that?
Well, we didn't ask you guys. What do you think it was? Are you going with the crowd?
You know,
Or
it's very hard after you hear all this lovely data to
it's true.
think anything else.
Got
You
it
should ask us last week, but then that wouldn't be right either, right?
No, I want to stop just short of giving it away, when Daniel reads a new case, because I think that, we know too much.
Did you think of it right away, Dickson?
Yeah, I did, actually. Yeah.
I thought Ascaris lumbricoides.
Ooh.
Just kidding.
Well, that's not bad a guess for a virologist.
No, that's not nice. That's very nasty.
Ooh.
No, I think Vincent at this point is a bona fide parasitologist.
Okay.
But
He's earned his worms.
So just to give people the wrap up, so this young man was referred to an infectious disease specialist who did, as Dickson points out, skin snips, not biopsies, right? So tempting to do biopsies because that's what people used to, but skin snips. And the small skin snips were examined, and very small worms with tapered tails were seen, and these were identified as Onchocerca volvulus microfilaria. Right. The boy went on actually to be treated. This was in days before doxycycline, so it was several cycles of ivermectin, and actually the nodules were surgically addressed. Mm-hmm. It's what I present these cases. It reminds me of when I was just at Columbia on Tuesday listening to a presentation of a case that turned out it was histoplasmosis in the lungs. And it was a case of a gentleman from the Caribbean who had acquired it there. And I asked there's a group of people that were there. And I said, now, if this case had been presented where they said, here we have an individual from the Ohio River Valley with something going on in his lungs, right?
You had Ohio River Valley fever.
There's certain catchphrases, right? So, I wonder when I present this case and I say, here's an individual and their job is to go to the river, so you have eye issues, the local river, when we get all these these facts, these little bits from the history, putting it together into the story. Hopefully we have like the story that I present, which is compelling. We have tender nodules. We have a lot of pruritus, so itchiness. We have eye involvement. We have the fast flowing river where the boy goes. Now, what other things? The itchiness, right? I think Wink brought up the issue of scabies. When someone is really itchy and they're from an area, what is another name for scabies, but the itch mite.
Right.
So, someone could be incredibly itchy. He brought up the issue is could that involve your eyes? And if you read most things, it'll say like, it really just involves below the chin on down. Now, that is true to some extent. And part of it is that there's a certain pH in the oils in the face, actually. But when you go to some of these impoverished places, you can actually develop a crusted scabies, which creates a thickened involvement of the scalp and actually can get involved in the eyebrows and can potentially involve the eyes. So, that was sort of an interesting presentation, but I don't think that would explain the tender nodules and the fact that, but no, so I'm glad that there were a few other thoughts, because if you don't have them in your differential, I think you can get yourself into trouble here. Okay. So no, this was Onchocerca volvulus. Another part that I really like about this case is that, I think we can get negative at times. The world's going to some place that we're not happy about. And this is a triumph. This was introduced, but as the WHO has announced, onchocerciasis has been eradicated from Mexico.
That's right.
It's great.
And that's great. This is not going to happen. You're not going to have people going blind out of Mexico from this. That is tremendous.
But not from Africa yet, right?
No.
Will it be?
Almost. It's eliminated from, I think, around 90%. It's still in, I think, Sierra Leone and Guinea. And some other, not Guinea, but Burkina Faso. I believe it's in Burkina Faso still. And you remember the difficulties when the Ebola outbreak occurred of getting compliance for just standard public health practice during the treatment of these patients. There were 2 countries that refused to allow UN workers and emergency medical workers to come into their country. And those are the countries that still have guinea worm, and those are the countries that still have onchocerciasis. Too bad. Yeah.
All right. Anything else, Daniel?
Oh, well, I think as we talked about, let's just I think, Dickson, you ran through the life cycle a little bit, but we'll just recap it quickly.
Yep, okay.
And then you can jump in if there's anything I should jump in on a couple little parts, which I think are interesting. So we'll start off the life cycle where the person acquires it. And I remember we talked about this before, and Dickson likes this point of you do not get this the way a mosquito. We've all discussed that, the way the mosquito does this probing. And a certain amount of, let's say in malaria, a certain amount is injected during the searching, but then it actually gets into a blood vessel. So these black flies actually do a sort of a cutting. They create a little bit of a wound, and then the larvae are going to crawl into the bite wound.
There's nothing subtle about the bite of a black fly.
Yeah.
Dickson always makes the hand scissors,
like scissors,
blades.
They are blades.
Cut.
That's true.
Yeah. And so then you have the process where the adult worms are going to actually mature, or they're going to mature into adult worms in the subcutaneous tissue, and you're going to get these nodules. And as mentioned, that's going to have something to do where in the world you're going to see concentrations in different areas, whether they're in the upper part of the body and the head, or whether the lower extremities, more so in Africa. And then what causes the symptoms? And there probably are a lot of more symptoms than we realize. There's a lot of itching as these are traveling through the subcutaneous tissues, the microfilaria. I mean, the adults are sort of hanging out in their little home. And there's an inflammatory process that can occur, and that is over many years going to lead to a progressive amount of eye damage. If you can get there early, you can stop that. But once it's already basically created an opacification, you're sort of too late. And the black fly, when it's doing this cutting, can actually ingest these microfilaria, which can then go through their cycle in the fly.
Right.
Later, after it's gone through its cycle, you can become infected again. But it is interesting, right? You've got the black flies in Africa, but what's going on here in America? Apparently, we have a very similar black fly that can.
That's right.
So
Exactly.
yeah.
So the skin condition has got a name that was applied to it by French doctors in the Cameroons and Côte d'Ivoire called peau orange, which is not a funny thing when you see it, and certainly not a funny thing when you got it. And it's excruciating itching, and it's unsatisfactory to just scratch because it doesn't stop the itching, unfortunately. And the skin loses its elasticity, and it loses its color. It loses the ability to protect against, the elements, basically. And it's really an awful disease at that point. And then there's something called hanging groin. Also, we didn't talk about it, but you can get an enlargement of lymph nodes in the groin area, particularly in male patients. And it's sometimes confused with elephantiasis, but no, it's caused by onchocerca, basically. The other thing to point out is that where the black fly bites, that's where the infection stays. The worms don't move an inch. They're couch potatoes. They don't go anywhere. They just enter the subcutaneous tissue, and that's where they stay. So quite interesting, actually. And then it makes you wonder how these worms actually derive their livelihood from the host. Forget the larvae at one point. Just think about the adults. And they wrap themselves up and assume a very small, compact area inside the skin. And they cause no basic harm. I mean, if that's all that occurred in this infection, nobody would get ill. But still, it's unknown as to how these parasites obtain nutrients, for instance, and get rid of waste products. And when you look at the process surrounding these nodules, it's hypervascularized. So what you've got here is a vascular proliferation, much the same as onchocercoma. That's what they were known as. I mean, these are non-proliferative nodules that have an exaggerated circulation to accommodate the exchange of nutrients with the parasite and the getting rid of wastes as well. And it very much reminds me of a multi-wormed occupation instead of a single worm occupation that you would find in Trichinella, which is about a lead into our paper this week. But this worm has a lot of interesting biology that we're still totally unaware of.
So why the cornea?
Well, they migrate everywhere. The microfilaria migrate everywhere. And the eye is contiguous with the subcutaneous tissues, and that's how it gains entrance into the eye. And it's really horrible once it gets there because there's a lot of damage, there's a lot of inflammation, and there's a lot of attempted repair, but scar tissue is basically the result, and it prevents the muscles from functioning, so you can't focus and you can't regulate the amount of light coming in, and therefore it's a horrible late stage disease in older people. Thank God it's on the way out.
Okay, should we give a book away?
Absolutely.
Yeah.
All right, we had 10 guesses, so I'm going to generate a random number between 1 and 10. Are you ready?
We are.
The number
Where's your... That's where the drum roll was.
The number is 5. 1, 2, 3, 4. Elijah is the winner.
Nice.
Is it Alicia or Elijah? I was going to go with Elijah.
Elijah is the winner. Elijah.
Nice.
Not only have you got a immune podcast today, but you've got a copy of the book. Send me your address, TWiP@microbe.tv. And if you are out of the U.S., I also need a phone number.
Uh-huh.
But if you're in the U.S., just an address. There you go. Congratulations. Are we going to do another one?
Yeah.
Keep doing it, right?
I think we should keep doing it.
Come on.
We'll do it until every person on the planet has a copy of that book.
Right.
How's that?
But they all have to listen. That's the other thing.
They do have to listen.
That's true.
And they have to listen. That way they can claim their gift, because we're not going to email and tell them. I want them to hear. Say, over on TWiV, we haven't heard from the winner of our book. And and someone said you should email them. I said, no, I want them to listen. That's the whole point, right?
And they hear, then they hear that they won. I was going to just, because boy, we're talking about onchocerca, and it's a great disease, fascinating disease. And I think, the ocular manifestations get all the play, right? River blindness. That's how I remember the blackness fly, right? But the skin manifestations are tremendous. I mean, the leopard skin, all this dermatitis, the hanging groin. I mean, the pruritus, the itchiness drives people just beyond. It's incredible. And then there's even, just to make a nod to nodding syndrome, there may even be a neurological disorder associated with just the amount of inflammation and the onchocerciasis infection, which maybe, we'll be honest, it isn't completely 100% cinched, but there's been a lot of connection, particularly epidemiologically, between seeing this syndrome and the areas where onchocerciasis is still prevalent.
Right. So imagine having to live with this as a chronic long-term disease and going to work every day. And the answer is you can't do that. So as this disease became embedded into the fabric of West Africa, people in wholesale numbers began moving away from the flight ranges of these black flies. And that cut them off from a resource that was life-threatening because, if you don't have any water, obviously you're going to not do too well. So
it can't be any worse than coming to work and seeing you.
I think we should let the audience decide that.
I'm just kidding. I actually am kidding. I joke. Sometimes you insult people, and that's a joke.
Methinks you just joketh too much.
Are you sensitive?
No, not really.
All right, let's take a look at this paper.
Yes.
Which was picked by Daniel in your honor.
I feel deeply honored.
Trichinella. It's in a journal called Parasites and Vectors, which I have not heard of before.
Well, it's always the first time.
It's always the first time, but it's a nice name, don't you think?
It is. I do like it.
And it's called Immunoproteomic Analysis of the Secretory Products of Trichinella, Pseudosporalis, adult worms, and newborn larvae.
Right.
First author is Yang Wang. The last author is Xiaolei Liu. And these individuals are from China.
Yes.
They are from the Jilin University, which is in Changchun, China.
Right.
They're all from the same place.
Yes. But there was a Harvard person there also.
Is there?
I recall. Yes, there was one Harvard person.
I just see here Institute of Zoonosis. I don't see any Harvard.
I saw in the contributions section of the article, I thought I saw some.
That would be the big H, right?
That's right.
Now, Dickson.
Yes.
This is not Trichinella spiralis.
Nope.
This is Pseudosporalis.
Correct.
Did you ever work on Pseudosporalis?
Myself personally, no, but I knew lots of people who did because when it was first discovered, it was discovered in birds. In fact, raptors. And it was discovered in Tasmania, of all places. And then in various other places. And if you looked at where it was discovered, it looked like the flight range of these migrating birds that indicated that this was something unusual, and we'd never seen that before. So
birds,
yeah.
And
that's right.
But it can also infect people, right?
Apparently, yes.
And how would you, by eating an infected bird? Is that how?
No, you could eat what the bird ate.
And what would the bird eat to get
Well,
sporadic?
the birds, sometimes they're scavenging. Sometimes they eat other birds, because a lot of hawks eat other birds because they catch them in mid-flight, as you know. You can actually watch that outside of our window right here.
Yeah.
We have red-tailed hawks that actually hunt pigeons out there, and so you can see them nail one every now and then. So the biology of Trichinella expanded the moment that molecular technology hit the sidewalk running. So now we could tell whether or not it was Trichinella spiralis, Trichinella nativa, Trichinella britovi, Trichinella zimbabwensis, Trichinella pseudosporalis, Trichinella, what am I missing? There's one in Papua New Guinea, Papuae. There are a couple of other species. There are 8 altogether. But I think there are 3 now that are non-capsulated Trichinella in the Trichinella genus.
9 different species.
Okay, 9. About the 9, oh, that's fine.
All right, so explain this. Do not induce formation of the collagen in the nurse cell. What does that mean?
So I spent most of my adult life trying to figure that out.
That's why I asked you.
And I never did figure it out, but at least I could typify what kind of collagens and when they rose. So Trichinella spiralis is considered to be a capsulated species of Trichinella. And during the formation of its capsule, as it sits inside of a muscle's fiber, converting a portion of it to its home, which is a complex process, not unlike some of the viral infections that you're used to looking at, where you get transformation of cells as a result of genomic rearrangements. Trichinella can actually affect the expression of host genes by secreting products into the milieu of where it is. And Trichinella can't make that collagen. We already know this. This genome does not contain a single gene for collagen type 4 or collagen type 6. And yet, the capsule that surrounds Trichinella is collagen type 4 and collagen type 6.
Very interesting.
How do that work?
Very interesting.
I went home every night and I
Did you figure it out?
No, I didn't, of course not. But I think it communicates with the host via its secretions.
Okay.
So that's why the attention has been focused on the secretions of various stages of this worm.
Secretions and excretions.
Well, because we can't distinguish whether or not it's getting rid of something or whether it's actually secreting something to affect a cellular function. So these gentlemen and these researchers, I should say, have all banded together. And in China, by the way, there's a lot of trichinellosis of various kinds, but mostly it's Trichinella spiralis. But they wanted to compare and contrast a capsulated form with a non-capsulated form so you can tease out what induces the capsule.
Can I ask a question?
Sure.
So you don't need a capsule, apparently, to be a successful Trichinella, right?
You do not. And that is exactly right. That is exactly. And in fact, with these species, the Pseudosporalis and the Zimbabweensis and the Papua, the larva can actually, once it matures and is infectious, can actually migrate up and down this muscle fiber. It culls out a much larger zone than the Trichinella spiralis and the other capsule-forming species. So it's got some big differences in its biology and therefore in its evolution. And there is a family tree that you can go back and look at and see which arose first. Quite amazing. So what we're looking at here is the secretions of the adult worms and of the newborn larvae, the larvae that are produced by the adults.
So let's go through quickly
the life cycle. Sure. So you eat a piece of infected meat, raw or undercooked.
And if you're a bird, they don't cook it because they can't.
Apparently not. At least we've never observed them doing that. So that's right. So this is a parasite, which is transmitted from host to host by carnivorism. There's no exception to that, by the way. It's all carnivorism. So in this case, the Trichinella pseudosporalis larvae, which are infectious, are eaten by the next host. They then go to the small intestine. They burrow into the columnar cells in the small intestine. And in doing so, they begin to transform into the next stage and then finally into the adult stage. So there are 4 larval molts. 4 larval molts, right. I believe it's a second stage larva when it's swallowed. And so it has to molt twice more before it becomes an adult. And then when it does, it has to mate. And then the mated females begin to produce live larvae, just as the Onchocerca filarial worm was producing them in the nodule.
Intestinal wall, right?
This is in the intestinal wall. And these larvae, these newborn larvae, as they're called, and actually we take credit for naming it, would penetrate into either a lacteal, which is the drainage system for fats, or into a capillary, or into a lymphatic. You have 3 choices when you're in the small intestine. The lacteal leads into the lymphatic, whereas the circulation from the blood stays apart from that until it enters the heart. The thoracic duct into the heart dumps all of the contents of the lymphatics into the bloodstream. It's all mixed into the heart and then distributed throughout the body. When that happens, these newborn larvae then are distributed in all directions, basically. And whenever they encounter a capillary, which is slightly narrower than their diameter, they get stuck. And this induces a thigmotactic response in the
Thigmotactic.
Right. You like that word.
Love it.
I know
Love
you
it.
do. So you can feel that you're being surrounded, and this stimulates the worm to extrude its spear. And in doing so, it can puncture out of the capillary bed into whatever tissue they're stuck in.
Right.
Now, this is the tricky part, no pun intended, but this
Trichinella part.
well, yeah, this is the part that's hard to envision, but it actually does occur. And that is that when newborn larvae encounter tissues other than striated skeletal muscle, regardless of the species, they're all heading towards that muscle cell. All right. Smooth muscle, no. Hard muscle, no. Although they will penetrate those two tissues. Once they get there, though, they have chemoreceptors. Mm-hmm. And they can say, where am I? They don't have eyes. They can't see, but they can taste. Mm-hmm. And they can taste. That's not a striated skeletal muscle cell. I'm sorry.
They move on.
They get right back into the circulation and overcome that thigmotactic response, and they get recirculated. Eventually, they all end up in the muscles. Right? Because that's 40% of the body weight of most animals is muscle tissue.
Yeah.
Okay. So now once they're there, what happens? Well, in the capsulated forms, within 20 days, they grow, of course, from a worm that's about 20 micrometers long by 7 micrometers in diameter to a worm that's almost a millimeter long and almost 36 millimeters micrometers in diameter.
Right.
And it stays at that diameter, by the way. The adult worm does not differ from that diameter, because if they did, they could never penetrate into a row of columnar cells because they would burst them all open. So this is a highly selected life cycle at every zone of contact between the parasite and the host. So the capsulated forms form within a 20-day period, and they elicit a circulation that goes around each capsule so they can exchange nutrients and wastes. And they stay like that until the host dies, basically. Mm-hmm. I mean, they can live at most mammals, if you're not talking about elephants, and of course they're herbivores, so they wouldn't normally encounter this infection. But if you're talking about most carnivorous animals, the average lifespan is 3 to 5, maybe some that live 10 years, maybe some bears will live just a little bit longer than that. But the fact is that whatever eats meat is subjected to this infection. Okay. And so it doesn't have to live longer than that, because when the host dies, it's consumed by other scavenging animals. And when that happens, and if there happen to be mammals, by the way, then they can be transferred from that dead carcass to the next set of hosts.
Got it.
And that's the end of that life cycle. Okay. So now it's also true, well, in Zimbabwe, guess what the definitive host is in this one? It's an alligator. It's a crocodile.
Crocodile.
It's a crocodile.
It's certainly a carnivore.
It is, but it's not a warm-blooded animal, right?
Nope.
It's a poikilotherm. It's an animal whose body temperature varies with the outside temperature. So that's not typical for Trichinella to infect animals like that. But yet here's the part of the evolution of this worm has included that group also. What are crocodiles distantly related to? Dinosaurs.
Really?
So where the hell did this worm actually come from to begin with? And if you look back in history, if it was around during the time of the dinosaurs, then one can understand how it made the jump into reptiles and then from reptiles into mammals. And so that's what you've seen, a progression of evolutionary development and spread. This worm refuses to take no for an answer. So it attracted my attention from the moment I heard its life cycle, which was not completely understood when I first studied this. We've taken a long time to come up with these details.
Got it.
So this is an interesting paper. Now you're going to go through all of the data here, and I'm going to reserve my comments until after you do that.
Now, they wanted to look at the secreted and excreted proteins.
Yes.
Right?
Yes.
Now, because as you said earlier, some of these must be the key to collagen or no collagen, right?
Right. And also, by the way, this is not a typical muscle fiber that this uncapsulated worm ends up living in. Yeah. It's got lots of bizarre cellular components that have nothing to do with a striated skeletal muscle. So this worm can still affect its microenvironment by its secretion. So it's still very interesting to look at in this sense.
Now, have these proteins been identified for Sporalis?
Well, that's what I spent some time in my lab on also. And a lot of people also did, because they're also diagnostically important, because they're antigenic as well. And therefore they're useful in knowing whether a host is infected with this or not. So yes, we've actually sequenced some of the genes responsible for the expression of these parasite products.
So they grow these worms in culture. All right. So they say they put them in RPMI.
That's That's not culturing. They're just maintaining them.
You know, they say culture, but you're
I
right.
know. It's a maintenance.
The maintenance. They're not growing, right?
No, they want to just allow these worms to secrete.
And they're secreting. So then they collect the supernatant.
Exactly.
And concentrate it. And then they do two-dimensional gel electrophoresis.
That's right. Proteomics. They're
which is an
proteomics.
old technology.
It is. I used to do that.
You know, you separate in one dimension according to
isoelectric points.
pH, right? And the other by molecular weight.
Exactly.
Basically.
Right.
So you can separate a lot more proteins than if you just did it in one dimension, say by size, right? So they see 400 spots just by staining.
That's amazing, isn't it?
All right. 400 spots. And then they take that and they transfer it to
a
membrane and do an immunoblot, a Western blot.
That's right.
Which we should call a far Western because it's many, many proteins. So, and they probe it with antisera that were made in pigs, I think.
Yes.
Against, just by infecting pigs. Right?
Pseudosporalis, this parasite, even though it's a parasite of birds, now remember, birds' body temperature is slightly higher than a mammal.
Yeah.
All right? And yet it can still survive inside of a mammal as well. Trichinella can't do that, by the way. They've tried to infect birds with Trichinella spiralis. It doesn't work. But Trichinella pseudosporalis, it works.
Yeah. So they have all these spots, and then by staining the protein, which is Kumasi blue staining, it just will stain all the proteins. And then they have the spots that react with antisera.
Right.
And they can correlate them. And they find 28 spots that they can correlate the staining with the antibody staining,
exactly.
Right? And then they cut them out.
Right.
And subject them to mass spectrometry.
Yeah.
Right, where they can put it in a machine and the machine
sequences the protein
will give you some sequence, and then you can look in a database, and they have a table here of 24 excretory secretory proteins.
24.
Right, numbers one. Actually, it says 28 here, I guess, but I saw 24 somewhere. It doesn't matter. It doesn't matter.
Yeah, that's right.
Yeah, and this is what you can do. 24 of the 28 were successfully identified.
Yes.
So now we have these proteins, and they want to know what they're doing. So they subject them to what's called gene ontology annotation, which is just use a software program that says, what are these proteins doing?
Exactly.
And they can get things like DNase 2, DNA binding, protein kinase activity, ATP binding, metal ion binding, et cetera, et cetera.
DNase activity. That's very interesting, I think.
So DNase is one of the ones they talk about. And they say most of the molecular functions of these proteins were DNase 2 activity and serine type endopeptidase activity. Now, Dickson, let me ask you.
Go ahead.
Serine type endopeptidase.
I think I have the answer for this one.
Does that mean it cuts at serines?
No, it means that the active site is a serine.
Hey, very good. That's excellent.
Did I remember right, Coach?
You did, and that's what you admitted last time that the guy was very impressed with. Yeah. And you actually, by doing that, you encouraged someone out there. So I think that's tremendously important.
Listen, you have to be humble in this one because it's a humbling... Science is a humbling...
You are humble, I must say.
I try to be.
You're a lot of other things, but you're humble.
At least I'm not a humbug.
You are humble. So DNase 2 is a well-known acidic endonuclease that fulfills a variety of functions from degrading DNA associated with apoptosis and dietary DNA to modulating host immune responses.
Isn't that interesting?
I didn't know that. Do you know anything about DNase 2?
I didn't, but I've heard this mentioned before because it's also found in Trichinella spiralis and in their secretory products as well.
Mm.
So it must be useful in some generic sense to ward off the evil spirit, so to speak, so this worm can have its way with us. They think of it as an anti-inflammatory compound that the worm uses, perhaps, but they're still guessing. Until there's a knockout worm that doesn't have
That's
DNA.
what I was going to ask you. Can we make knockout worms?
You probably could.
CRISPR?
Yeah, you could.
Yeah. Yes, Daniel.
Yeah. Oh, I was breathing, but yeah. So you can.
He was breathing.
No, but it was a thoughtful breath. And I think we may have presented this paper on a prior TWiP. But so you can use the kind of stuff I use in the lab, where you use lentiviral based vectors to introduce using the VSVG envelope to introduce genes or, basically something that will block gene expression in Trichinella. So, yeah, this system is one that you can manipulate.
Right.
I was going to make an interesting point, I think, before we go too far, is that so when they do the initial, right, they do this initial ES far western blot, we'll call it,
Yes.
and they run out these 400 spots. So these are the 400 spots which are all of the different ES proteins. The second thing they're narrowing it down to is not necessarily the most important for the collagen or the other things. They're isolating which are the ones that the pig has a immune response to.
Correct.
Exactly.
And
Correct.
so these are what are the most important from the immune system of the pig, not necessarily what are most important from the construction design.
Totally correct.
Of Trichinella.
Because they have it in the back of their mind to use these as immunodiagnostic tools as well. The only thing that I'm a little dismayed about is the last two words in the title. I don't see where those were analyzed.
Newborn larvae.
Yeah, where I think when they take the adult worms out, they're taking them out when they're fully gravid, when they have newborn larvae inside of them. And so when they collect the secretions, they're not sure which ones come from adults and which ones might come from the newborns. So they had to put the title to reflect both adult worms and newborn larvae, but they didn't distinguish between those two things, which they could do simply by separating newborn larvae from the adults and then collecting their secretions. But that was my only quibble with the article. I mean, this is a generic approach to saying what's possible first with regards to the secretions of these parasites, and then which ones are important for which functions. That comes next. And by guessing at lookalike molecules that have already been described and by trying to place them into context, it's very difficult to get a clear picture of how these are being used unless you could tell when they're being used. And that, I'm sorry to say, was the basis for my last grant that I didn't get funded for. It's just, we have a way of actually synchronizing the infections, and so we could stop them at any moment and say up to this point which ones are secreted, up to this point which ones are secreted, and correlate that with cellular changes to see whether or not we could make an association between the secreted protein and the host change that it might induce. And I thought it was, we may have just been submitting at the wrong time in terms of the kinds of grants that were being funded, but I was somewhat disappointed. That's not putting it mildly. I was extremely disappointed that it wasn't funded because I would have loved to have seen the answer.
But I like this approach. I mean, you could use the approach they have here and say, okay, we're going to be doing these far western blots at each of these different time points. You've got your different spots and seeing when different ES products are coming up. And the interesting thing, too, is here, they're only focusing on 28 of these and looking at the immune response.
You've got 400
that leaves 400 minus 28. And that leaves,
Yeah. And imagine, now, Vincent, when you, on TWiP, because we're hosts on TWiP together, when we talk about viruses and I quiz you as to, that's a pretty big genome, for instance, a mimivirus. How many proteins does that encode for? And you say something like 200 or 300, and then maybe you come down into it. There are viruses that only have 3 proteins, right?
Some have 1.
Some have a single protein.
And some have none. We call them viroids.
Okay. Look at this parasite. It's got 400 different
secreted proteins.
Secreted detectable proteins. It could be others that you're not even seeing.
Sure.
And what the heck are all those things doing? It's amazing.
One of the biggest genomes is an orchid.
Yes, indeed it is.
It takes a lot of genes to be beautiful.
It does.
Some of the other ones here include a venom allergen.
Yes.
Which they say might help adult worms evade host immune responses.
That's right.
But yes, if you're interested and you're working on this, you have a gold mine here. You can start
You do.
knocking these out and seeing. But you need an experimental system, of course.
Yeah, it's too bad they can't grow this in vitro, because if they could, then they could work a lot easier.
You couldn't grow Trichinella in vitro? No. What did you do? Grow it in mice, right?
Yep.
So you could put these in. Can these infect mice, the Pseudosporalis?
Oh, sure.
So they could use that.
Yeah, so the big deal about Pseudosporalis, as opposed to Spiralis or any of the capsulated versions, is that there is zero inflammation around the affected portion of muscle cell in Pseudosporalis. None. There's nothing to indicate that there's a worm there. There's no neutrophils, monocytes, T cells, nothing.
Yeah.
No eosinophils, nothing. Whereas in Sporalis, there's an intense inflammation around each nurse cell. Remember, we've read a paper by Appleton that suggests that the eosinophil fusion helps the parasite mature. This is just the opposite. This parasite has a very potent immunomodulatory pathway, which it controls the host's response to.
Some of these proteins must be involved, right?
Exactly right. So if you could knock just those out, you could probably knock the parasite out of the host.
Yeah, very interesting. So I think you ought to get back into this, Dickson.
I am into it. No.
Start writing grants.
No, you know what? No, let's not go that far. But we can certainly review these papers and offer suggestions as to their next approach.
I think you would be a wealth of information for these. They should consult you.
I would love to talk with them sometime about this because it's never disappeared from my radar screen as being my all-time favorite parasite group. I love Trichinella, just like you love poliovirus.
I love viruses. I love all viruses.
I love Trichinella. It was amazing.
You need to have a Trichinella license plate.
Oh.
No, you don't want it. Too many letters.
T-Sporalis would be fine.
T-Sporalis, yeah, that would be perfect.
Yeah, we could do that.
Oh, that would be awesome.
Yeah.
Just do it so I can take your picture next to it.
Well, you remember what the logo is for our podcast, right? It's the nurse.
Nurse cell.
We use the nurse cell as our logo.
I'm going to make you a phone case with a nurse cell on it.
I'd love it. I would love that. Daniel, thank you for picking this article, by the way. I appreciate that.
He's very touched, Daniel.
He is.
Well, yeah, I was thinking of you, Dickson.
Oh.
I'm touched in many ways. And that's only one of many ways.
Dickson, do you have a hero?
Yeah, I do, as a matter of fact.
I know you were getting ready for it.
Thank you for asking. So we're going to go through this book systemically and systematically. So the next, and you're going to like this one, Vincent, because he is
Italian.
He's an Italian.
Yeah, I knew you were going to say that.
His name is Amico Bignami.
Amico Pignani?
Bignami. Bignami. How would you pronounce that?
Pignami.
Pignami?
No, with a B as in boy.
B.
Best.
B-I-G.
P-I-G-A.
No, B.
B. A-B. A-B.
And it would still be Bignani.
Bignami.
It wouldn't be Bignami, okay? The G is soft.
Very good.
Bignami.
His dates are 1862 to 1929.
Right.
Amico Bignami.
60 years old. Not bad.
Not bad. Okay, here's what it says underneath his portrait. Amico Bignami collaborated with Ettore Marchiafava and Giovanni Battista Grazzi. Now, Giovanni Battista Grazzi is a famous guy.
Yeah.
Because he's given credit for having discovered that Plasmodium falciparum is transmitted by Anopheline mosquitoes. Mm-hmm. And he played a role in that discovery. So he says, collaborated with these other two Italian workers in 1892 and elucidated the main clinical features of malignant malaria or Plasmodium falciparum. Bignami also showed that the crescent stage, the comedosite, did not cause illness. In 1898, Bignami and Grassi worked with Antonio Dionisi and Giuseppe Battistanelli to prove that Anopheline mosquitoes were the transmitters of human malaria. Now, the significance of saying that is that the Nobel Prize for malaria research, proving that malaria was transmitted by mosquitoes, was not awarded to these gentlemen. Mm. It was awarded to someone else. And we will get to that. And I don't want to spoil it for anybody.
That'll be another.
But it's a raging. They have never forgiven each other. The Italians and the English have never seen a peaceful moment of discussion with regards to who was actually worthy of that award. Because unfortunately, the Grassi group did not get recognized.
All right.
And the other one, of course, discovered it in Canaries. So.
Okay. Thank you, Dickson.
You're quite welcome.
All right, Dan, you have another case for us?
I do. And I hope people enjoy this case. This is a case where people from around the world were actually brought together to sort this one out. It's actually it was a physician that I got to know in Lima who ultimately got involved and came up with the diagnosis. But this is an individual in his 30s, was a male, and he had presented to his physician with stomach upset. He was saying,
is in Lima, right?
No, we're not there yet.
Okay.
So, but it was actually a physician that I met in Lima who was ultimately involved in the isolation of the pathogen or the characterization or identification. But here we have a gentleman. He's a male. We don't know if he's a gentleman, but he's a male in his 30s. And he's presenting to his physician with stomach upset and an uncomfortable feeling in his legs. Okay. Initially, he didn't think anything of it, but then he noticed motile objects in his stool. Oh. And he became quite concerned. We get a little more history now. This individual is up in Vancouver in British Columbia. He was eating a significant amount of salmon. We're not sure if that's relevant. He reports that all the salmon, though, was either dried through a smoking process or had been marinated in this dill marinade. So he was concerned, and he actually, this is like one of the things that happens as a physician, he brought in these objects to his physician.
Were they motile when he brought them in?
Yes, they were.
Outstanding.
So he's bringing in these motile objects to his physician. Mm-hmm. And that's all I'm going to tell you at this point.
That's cool.
That's
cool.
Very cool.
I like that.
And he's symptomless, basically.
Well, he was, it's interesting. He was
except for the psychological effects.
Hey, it's stomach upset, no?
Yeah, it's stomach upset, and he felt an uncomfortable feeling in his legs. Now, whether that's related or not, we can discuss at the next juncture. I should just say that there's nothing exciting on his physical exam. This is just this very interesting story.
How long has he had this problem?
And not for a very long period of time.
And it just came on, this stomach upset and leg business came on suddenly, right?
They're sort of vague. They're not, dramatic. But what really sort of, made him aware that he needed to see a doctor, or what motivated him to see the doctor, was these motile objects in his stool. People don't like when they look in their stool and they see things.
No, they don't. No, they don't.
People look in their stool often?
You'd be surprised.
So that's a good question. I was discussing this morning with another infectious disease doctor. That's what we talk about, and because I'm
over coffee and donuts.
Well, I was letting them know I'm in a good mood because I'm going to be heading back to Panama in March.
Oh yeah. I'm
going to be doing more work with the floating doctors, and there's actually a CME that I'm going to be teaching at nice. Down there in March. Nice. And it's nice. There'll be lectures. There'll be a couple days of actually remote clinic, hands-on field work, so people get a chance to learn this, I guess the science, the medicine, but also do some work. But I was saying when I was down there last, and I do this whenever I go to the, other parts of the world, I will joke a little bit with the kids when the mother says, "Oh, they're here because I'm worried about worms," and I'll ask the little kids, so are they big? Are they small? And then when they start giggling, I say, so you shouldn't know. If you're really using the outhouse like you're supposed to, they should be falling into the abyss.
Exactly.
So what are you doing looking at your stools? You must not be going in the right place.
Interesting. Right. Very interesting.
I remember my first visit to Europe, and I went into the men's room, of course, or the bathroom, I should say, because it was in my hotel room. There was a little platform onto which my stool fell. There was no puddle of water like there is in a typical American toilet. There was a platform. It was dry. It was porcelain. And it allowed you the luxury of turning around and looking to see if there was something in your stool or not before you flushed. And I thought, wow, that must have its origin somewhere back in the discovery of parasites and then tapeworms and then large things like ascaris and that sort of stuff, which would, of course, allow people to self-diagnose and bring things in.
This brings up an interesting point.
Right?
I've noticed that in general, in Europe, the toilets have very little water in them. Yep, whereas here in the U.S., we have quite a bit.
This is true.
And that is probably an issue of odor, right? Because if feces fall into water, the odor is more suppressed than if they're sitting out on a porcelain shelf, right?
Yeah.
But you made me just think of that. Thank you very much for that.
Not at all. This is what infectious disease people do when they sit around and talk.
No, but people, that's often a complaint. People will say either the frequency has changed,
sure.
color, or people, people are aware that there's a relationship between their what comes out and what might be going on with their health.
Yeah,
Exactly.
it's good. It's good that they noticed this.
Very cool.
All right. So with that, we will move to a few emails. And the first is from Andrew. Hello, fellow parasitologists. I must say I'm hooked on your podcast. The level of knowledge that you bring to all these organisms in which I studied in college and work with almost every day is tremendous. I am a medical technologist in a large clinical diagnostic microbiology lab in Boston. My main areas of interest are parasitology, bacteriology, and mycobacteriology. I would be the guy in the parasitology lab who would be identifying this worm for the doc, so I figured I'd give it a shot.
Right.
I love my job. It's always fun because every day brings something new and unexpected.
Here, here.
I'm the guy at the microscope, not at the patient's bedside, so I don't see the clinical manifestation side too often, so please excuse me if I am incorrect on some counts. So this is a late entry in the previous
Right.
one, which was Trichuris. I believe that was the answer, right? Not last time, but the previous time, Daniel? Was that right?
Yeah, I remember this time.
So-
Yeah, it was the colonoscopy, right?
Yeah.
Yes.
Yeah.
Yes.
Yeah.
So I'm going to skip most of this because we've gone over it. And I just go to the end where he says, thanks for making me do some research to discover some new things. I always appreciate learning. Keep up the great work with the podcast, and I promise I'll keep listening. Your friend Andrew. So after his name, Daniel, he has MLS parens ASCP parens. So tell us what that means.
I'm not sure, actually. Let me see here. So the American Society of Clinical Parasitologists, that's the ASCP, and MLS, I don't know, is that a master's
In laboratory sciences.
in laboratory science? Okay, yep.
Medical laboratory scientist.
Yep.
I'm sorry, I didn't mean to put you on the spot. I thought you would know.
No, that's no.
The three of us worked it out.
Yeah, no, I tell people this is that, people often throw these acronyms, and it's a challenge for me because I've sort of, I'm in each different arena. So, is it, pediatric critical care medicine? Is it pulmonary critical care medicine? Is it, I get thrown these acronyms and I'm not really sure like which arena they're referenced to.
So that one and the next one from Chris came in late. Chris had an email in this time, and Chris is in Athens, Georgia, where he says the chance of snow is still far, far away. But then in today's email, he said, we may get some snow.
Right.
Okay.
All right. Assuming you're still giving them away, here's to the off chance of adding a new parasitology textbook to my shelf and arousing the envy of my coworkers. All right, Dickson, why don't you take Trudy's?
Dear TWiPers, I have a question regarding related to parasite exposure dosage. When it comes to other microbes, such as bacteria, for example, I usually think that the likelihood of someone developing symptoms of infection depend on the amount of the microbe the person has been exposed to. I think that this is generally also the theory behind washing produce. Behind washing produce. You will never get rid of all the dirt, soil, or poop, but the idea is to get rid of most of it so you don't get sick. So my question is, can washing produce help us prevent infections with parasite embryos as well? I think it's interesting that the lady in TWiP 142 developed symptoms from having just one worm. Is that all it takes for some parasites? On the other hand, it seems to me that it was unclear whether the anemia was related to the whipworm infection. Please clarify if possible. Well, indeed, multiple exposures increases your chances for developing clinical symptoms from any of these. Virtually no nematodes except for Strongyloides and its relatives can reproduce once it gets inside of you, so that you're pretty safe. When you swallow one Ascaris egg, you're going to end up with one Ascaris. You swallow one Trichuris egg, you're ending up with one Trichuris. Neither one of those parasites by themselves could cause harm unless the adult Ascaris would migrate, like, say, into the ampulla ovata or into the common bile duct. In that case, a single worm could cause quite a bit of damage. So it depends on the worm, and it depends on where the worm is located. And I think you could say the same thing for a single fasciola that got aberrantly located in your brain. I don't want to sacrifice any of my brain to a parasite, no matter what parasite we're talking about. So I think you'd have to go through them one by one and sit down and just look at the possibilities and connect it with the clinical symptoms.
But sometimes one is enough, right?
Sometimes one is enough.
Dickson,
but like with hookworm, for instance, it's not
enough, sure.
You need hundreds to cause symptoms.
Now, Dickson, the one transmitted on watercress.
Yeah.
Is it Clonorchus?
No, that's fasciola. That's fasciola.
Now, would washing help?
Great question. And of course, no, because the Metasicaria is on there with a glue. It's basically, it sticks to anything and then it does not wash off. So
is that the one where people do something?
No, that's another one, but it's a little bit like that. That's Fasciolopsis busci. In water.
Excuse me.
You asked me the question, I'll give you the answer, but he doesn't like my answer. I can come up with another answer. Opistorchis viverrini. No.
Oh, okay.
No, in this case, the Metasicaria of all of these trematode infections cling using a special substance, which I guess if we knew what it was, we could invent a new class of glues.
So you can't wash it off.
You can't wash it off. But if it's an egg from, let's say, Ascaris or Trichuris or a larva from hookworm, the answer is you could possibly wash it off.
Yeah.
Possibly. Yeah. You can't wash off microbes, though, because there's too much interface between the parasite and the substrate to be able to just use simple water and get rid of it, unfortunately.
Daniel, do you remember your case on jumping salad?
Yes.
Now, could you wash that?
You couldn't wash it, but you could sieve it.
The interesting, I mean, a good way to, if you like to eat things that are not cooked, is soaking things, right? You could soak things in this dilute bleach, which is what people do with their fruits, vegetables in a lot of parts of the world. Actually, when my parents they had a place down in Florida, and a woman from the islands was helping my mother. And my mother was saying, "Oh no, you don't actually need to soak everything in bleach. You're here in the United States, but I'm not sure that's true anymore." So, because we import a lot of stuff from a lot of parts of the world, and as we repeatedly see, there can be transmissions. Mm-hmm. And so, soaking things in bleach, and then rinsing it off can work. As Dickson went through, you could sort of go through each one's where can one be a problem. So Ascaris with aberrant migration, schistosome if an egg ends up in the CNS.
Right.
In most cases, the rule is it's based upon worm burden, not just one.
That's right. Whereas in protozoan infections, they more resemble the microbial world. When you have one and then it starts to multiply, the symptoms start to increase as the numbers of parasites increase.
Yeah, and with a lot of infections, even getting outside of parasites, viruses, bacteria, there usually are inoculums. We talk about low inoculum infections and high inoculum infections, like color. You usually require a pretty big dose to get sick, but other things like let's say norovirus, for instance, or Shigella, these are pathogens you don't need such a big dose.
Great. So there's a PS to this, of course. PS Trudy writes, I'm always looking for an excuse to visit New York City, so I would love to be in the studio guest. I would love to be in the studio as a guest on TWiP. And are you kidding me? I also have a book for Dickson to sign. Last time I was in the studio, I had not yet read West Nile Story, even though I have a PhD on the topic, and I am a huge fan of Dickson Despommier. Well, so am I actually. No, that's not right. The shame. This has since been corrected, and I am looking forward to my next opportunity to meet Dickson. So I too look forward to meeting Trudy. Yeah,
we can tell you, we don't schedule this very far in advance, unfortunately, Trudy. TWiP, that is.
Yeah. Well, we have a TWiP every Friday.
Yeah, so that's she came for a TWiP.
That's right.
But we'll be in touch. We'll figure something out that you can.
Right. And where is Trudy located? I'm sorry.
Georgia.
She's in Georgia. Right. Okay, then.
Daniel, can you take Sarah's, please?
Sarah writes, greetings, esteemed triple TWiP doctors. I write to you from an uncommonly sunny Glasgow, Scotland, 10 degrees C. First email to TWiP, my favorite podcast for years now, and I'd like to express my sincerest thanks for keeping it up and always being so edutaining. I love the introduction of Daniel and his case studies. I've been guessing silently from the start, but I'll make an effort to start sending in guesses from now on. I also really like Dickson's little heroes, profiles, etc. of parasitism segments, and I thought I'd send some cool links and a suggestion in relation to them. I've just become the chapter leader for Science Girl Glasgow, a group of Scotland-based science communicating gals, guys, and babies. And over the past year, we have introduced a new activity that we take with us to events like science festivals, schools, care homes, etc. It's called Unsung Heroines, and we have centered it around UU2. Check out the links below, and there's a bunch of links below. A bit self-serving, but I thought I'd try for a TWiP bump. Always it would be cool if Dickson made one or more of his little segments about a female scientist working with parasites. I'm sure there's some, and I'd love to know more. And I'm going to actually say that's an interesting thing. When you go through this list.
We don't have any in our book.
Yeah, we gotta correct that.
I can scour the horizon for them and come up with a number of them. But, it was a very, very closed societal loop. The Germans came up with the parasitism sciences first, and then it spread to the United States, and it was a male-dominated science for many, many, many years. And it took a long time to even have a president of the American Society for Tropical Medicine and Hygiene as a woman. And it's too bad, because really there are lots of very talented women out there doing science research in those fields. So I will do my best to correct this gender skewing and to come up with something more equitable.
Yeah, the outgoing president was actually Pat was a woman.
That's right.
So not sure who the incoming one is. I think it's a man again. And as Sarah continues to write, huge thanks from myself and Science Girl for your science communication efforts and a possible TWiP bump. And if I don't get a guess in before Christmas, I'll send my season science greetings via and then there's a hashtag elf on a science shelf now. And there's a December 1st Science Girl Glasgow Twitter advent calendar link. Happy holidays, Sarah.
Fantastic. And back to you.
What does that mean?
It means the same to you.
Oh, got it. I thought you were pointing at that.
No. I'll point at too.
He's gonna make me read extra emails, right?
Just two more, both from Anthony.
Right.
I was thinking about trichurus transmission. Dogs defecate most anywhere and mouth, chew, swallow most anything. How they catch whipworm is clear. With the use of night soil and contamination of vegetables, it's easy to see how people become infected. With cats, it's not obvious. Cats defecate in particular spots. How would they consume dirt from there is not easy to see. Do mice digging up and consuming cat feces serve as fomites? Or might there be vertical transmission / spread?
That's a great question. I don't know what the species is in cats. Does it Trichurus have a species specific to cats? Because there's one for sheep, there's one for pigs, there's one for humans, there's one for mice, Trichurus murus. I don't know if a cat has a specific species of Trichurus.
Let's find out. Is there cat-specific Trichurus? Trichurus serrata.
There you go. So cats probably would eat a mouse whose feet were contaminated with the eggs of Trichurus serrata. And that's how I would guess it gets in. Because they don't lick their food. They just swallow it. They just chomp down on it and boink, down it goes.
Okay.
Yep.
And he also sent a site. If you think it looks good, it might rate a mention, and it is a course site from Penn, Veterinary Parasitology.
That's a great school for that, by the way.
Lovely.
It's a vet school course.
Resources. They have life cycles, glossary, previous exams, and case studies. Well, look at this case study.
Nice. Rufus
is an adult mixed breed dog that has been exhibiting intermittent symptoms of otitis. Look at all these cool. He frequently shakes his head and paws at his ears. On examining Rufus, you detect a significant amount of a flaky brown material inside the pinna of each ear. Perceptive veterinarian that you are, you use a swab moistened with mineral oil to pick up the specimen seen at the case study, and you see photograph below. Looks like some kind of an animal to me. Is it scabies?
I'm not looking,
Tick?
so I can't see.
I know, but anyway, it's an animal.
Has it got legs?
Yeah, it's got legs.
How many?
1, 2, 3, 4, 5, 6.
Sounds like an insect.
It is some kind of an insect. Then they have questions.
Or a larval tick.
Which case study is this?
Case study 3, arachnids and small animals.
Arachnids and small animals.
This is lovely. They have old exams.
So it might be a mite. It might
index
be a
to techniques.
It looks a lot like a mite, actually.
Mite, yeah. They don't have a link to TWiP. Shame on you. Because we don't talk a lot about veterinary parasitology, do we?
No, but we talk a lot about the involvement of animal vectors and reservoir hosts and things of this sort. So I think that it fits in very nicely there. I used to know the guy who ran that department. His name was Lawson Soulsby. He later on retired and went back to Cambridge to become the dean of the vet school there.
Wow.
And was eventually knighted and became Sir Lawson Soulsby. And if you knew him, he was a hoot. You could go out and spend all night with him. And, he would, along with his colleagues, and I considered myself a junior colleague of that group, we used to go to the meetings together and appear to be bleary-eyed the next day at the sessions. And it was hard to remember what was said sometimes because of all the ribaldry we were having the night before. Yeah, he was a good man. He was a very, very good man and very erudite and extremely distinguished as well. So he really was the best example I can think of someone who was a veterinary parasitologist, Lawson Soulsby, and I think was one of the founders of that department.
The other point from Anthony, perhaps there might be a feature on parasites that are not. Here's one. A dentist once told me that the root canal procedure was developed in the belief that the nerve was a worm. Gives a link to that.
Look at that.
The existence of toothworms was not disproven until the 18th century.
Right.
You know any others, Dickson?
Yes. In fact, they're called mucus casts, and they're extruded in stool. And they're brought to the lab, and you take one look and you know darn well that it's not a worm because the surface is uneven. And worms' surfaces are smooth. But mucus casts are very misleading. You can sometimes mistake them for tapeworms and things of that sort.
Daniel knows some, too.
Of course he does.
All right.
Come on, having been to Panama, I'm sure.
Parasites that are not.
That's right.
All right. Daniel, you still there?
I am still here.
Your video's frozen, so we don't know.
Frozen in time.
That's okay.
Maybe that's good. Hopefully, I was in a good pose when my video froze.
Well, I wouldn't.
You're looking
it's not particularly flattering from here, but
okay. All right.
All right. That's TWiP 144 microbe.tv/TWiP, Apple Podcasts, and wherever finer podcasts are available on your app. Just subscribe. And to those of you who support us on Patreon and by other means, we are very grateful for your support. If you don't, please check it out, microbe.tv/contribute. We'd love you to help contribute financially if you like and are able to do so. And please send your comments, questions, case solvings at TWiP@microbe.tv. Daniel Griffin is at Columbia University Medical Center and parasiteswithoutborders.com. Thank you, Daniel.
A pleasure as always.
Dickson Despommier is at thelivingriver.org and also parasiteswithoutborders.com. Thank you, Dickson.
Had a great time.
Did you?
I did.
I'm glad to hear it.
Yep.
I'm Vincent Racaniello. You can find me at virology.ws. The music on TWiP is by Ronald Jenkins, ronaldjenkins.com. Thanks to ASM for their support of TWiP. Been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.